background and motivation to the nitrogen cycle. Nitrogen fertilizer are inefficiently being used, which causes the atmospheric nitrogen to be excessively absorbed into soil. Our main goal in our MATLAB script is to collect and organize sets of data that are already existing for additional research and make an experimentation for an outside group and find the most efficient way to make improvements. The program is set to allow users to read, alter the data, and utilize their advantage. By doing this, the user can find ways to reduce the excess accumulation of nitrogen fixation. In our script, our group created a code that allows the user to access the data for a specific time, diameter class, plant types, biome, and route. Depends on the user's selections, updated graphs will appear with the new included data.
My name is Michael Heckman, and I will be talking about the first section of the code, which looks at biomass levels for all the samples of the years 2013 and 2014 based on the data set that we had. This program allows the user to select a year between either 2013 or 2014 to look data at, and after they select a year, the code will output which plant had the highest biomass through that year, which one had the lowest biomass through that year, and also it would output their corresponding carbon and nitrogen percentages, as well as their carbon to nitrogen ratio. Their carbon to nitrogen ratio is just calculated by taking their carbon percentage and dividing it by their nitrogen percentage, and it gives a ratio. Before we allow them to see that data, there's got to be some data validation. So throughout the entire first section and the second section, there's data validation. But for the first section, we data validate by making sure that all values are loaded in correctly, and we set the variables and make sure that they're easy and clearly understandable. For menus, we make sure that you're not able to exit out of them, so the user is forced to click an option between 2013 and 2014, or any of the menus, they're forced to click an option. For the biomass levels, once it displays the lowest and the largest, there is a warning for when there is a biomass that's less than or equal to 0, and we replace it with 1 if that is true in the case. And for every value that has a NAN, which is not a number, we replaced it with the -1 for temporary purposes. And the -1 also means that it's an untested data sample. After displaying the largest, smallest, and their ratios for biomasses, the code then proceeds to graph plant number for the x-axis and biomass for the y-axis. And after plotting the data for this, it then proceeds with another menu and asks the user if they would like to view data based on a type of plant. And once this menu is popped up, then again the user is no longer allowed to click the X on the menu, because it will just reopen the menu because there's a while loop there. So they are forced to click either yes or no. After they choose an option, if they choose yes, then it continues on to the next part of the code, and it allows them to choose which type of data they would like to view. And the options are understory, leaves, twigs, and above ground vegetation. These are just the different types of plant samples that were found in our data. Once the user chooses a type of plant they would like to view, it then displays all the data for that plant within that same year that they chose on the first menu. It will display the plot number, the biomass for all the plants, the percent nitrogen for all the plants, the percent carbon, and also the carbon to nitrogen ratio. It will also display which habitat the type of plant was located in. And if there are any negative values in the output, it just simply means that the data was untested for that specific part. After the user is satisfied with the results, another menu is popped up with also data validation, preventing them from to click out of it, and it asks them if they would like to run the program again. If they choose yes, then it runs through the program again, same menu, same everything. If they choose no, then the program ends, and it tells the user the total number of times that the program has run.
The next part of the program uses the data from two different spreadsheets of the raw Excel dataset, since the data from these two sheets correspond to each other. It starts off by loading in the data using the XLS read function. For this part of the data, there were a total of 100 different plant samples, and the root length was measured for each sample. This root length data is the main portion. We started sorting the data by creating a separate variable that contained all of the root length measurements for the samples. Since there were 100 samples and each sample was measured a total of 14 times, this variable for the root length data was a 100 by 14 matrix. We also established a variable for the measurement dates, and this was a 1 by 14 array of day numbers. Now, each plant sample is identified by 4 different distinguishing aspects: the habitat, the plot number, color class, and diameter class. So we sorted each of these into their own variables. Each sample also had a corresponding value for percent nitrogen, percent carbon, and carbon-nitrogen ratio, so a variable was created for each of these 3 values. After sorting the data, we did data validation. We made sure that values in these variables were the correct types of values using the isString function. We made sure that the habitat and color class variable contains strings instead of numbers. We also made sure that the dimensions of the variables agreed with each other. Each of the variables should have 100 values since there are 100 different samples. Now, in the raw data set, some of the plant samples were not tested for percent carbon, percent nitrogen, and carbon-nitrogen ratio. So the program asks if the user would like to see which samples were not tested for. Program finds these samples using the function isNotAnumber, since the values that were not tested were loaded in as not a number. If the user chooses to see these samples, the program will identify each sample that was not tested by outputting the habitat, plot number, color class, and diameter class of that sample. Next, the program asks if you want to add in your own values for these non-tested samples. If the user chooses yes, you'll then be prompted to manually input a value for percent nitrogen and percent carbon, and the program will then calculate and output the carbon-nitrogen ratio using these values. This is done for each of the non-tested samples, and the program then updates the associated variables with these new values. Another side note about the program: each of the menus that pop up will keep popping up if the user exits out of them. So the next part of the program is the option to view the root length data for a specific sample. The program asks if the user wants to view data for a specific sample, and if the user chooses yes, then the program will ask the user to identify the specific sample of interest. This is done by using a series of menus, asking the user to choose a habitat, plot number, color class, and diameter class. The series of menus required a nested for loop, and since there were 4 menus, there were 4 for loops. This part of the program could have been done without using for loops, but it would have taken hundreds of lines of code, and we would have essentially had to hard code the process for each of the 100 samples. Based on the choices of the user, the program will then find which sample the user is trying to look at, and it will then display the root length data for that sample. The root length data is displayed as a plot of root length versus day numbers. The title of the plot is generated specific to the sample being looked at. Displaying the data as a plot serves to allow the user to easily read the data and even draw conclusions and relationships based on the trend of the plot. The program also outputs the percent nitrogen, percent carbon, and carbon-nitrogen ratio associated with the selected sample. Lastly, the user is asked if he or she wants to look at a different sample. If the user chooses yes, then the program will repeat starting with the menus for identifying the specific sample, and if the user chooses no, the program will end there.
Humans have been affecting the nitrogen cycle for many years in many different ways. About 450 million metric tons of fixed nitrogen are made each year using a chemical method called the Haber-Bosch process. In this process, N2 is reacted with H2 at high temperatures. Most of this fixed nitrogen goes to make fertilizers we use on our lawns, gardens, and agricultural fields. In general, human activity releases nitrogen into the environment by two main means: combustion of fossil fuels and also the use of nitrogen-containing fertilizers in agriculture. Both processes increase levels of nitrogen-containing compounds in the atmosphere. High levels of atmospheric nitrogen are associated with harmful effects like production of acid rain. Also, when artificial fertilizers containing nitrogen and phosphorus are used in agriculture, the excess fertilizer may be washed into lakes, streams, and rivers by surface runoff. A major effect from fertilizer runoff is saltwater and freshwater eutrophication. In this process, nutrient runoff causes overgrowth or a bloom of algae or other microorganisms. Without the nutrient runoff, they are limited in their growth by availability of nitrogen or phosphorus. This code allows the user to analyze different samples of plants in different locations in order to find percent nitrogen and carbon. Doing so will allow the user to understand how much nitrogen to use in a fertilizer or how to use fertilizers more efficiently. Since the code outputs this data for many different samples in different locations, the user will be able to produce a different fertilizer for every plant type in order to control the release of nitrogen into the earth. A major limitation would be that there are thousands of different species of plants in the world, and this program may not be able to accurately analyze every species known on Earth. There may also exist some errors in the actual data used in the program, even with the data validation. These errors may come from possible mistakes in the actual scientific research done on the different types of plants.